Hello, and welcome everyone to Bio-Rad's webinar series on Droplet Digital PCR. My name is Mohini Patil, and I'm the Global Product Manager for Droplet Digital PCR at Bio-Rad and your host for today's webinar. First, let's discuss the webinar logistics. The webinar will last approximately 1 hour. Dr. Dawne Shelton will present on Droplet Digital PCR and its applications for molecular pathology and liquid biopsy for about 45 minutes. Following the presentation, we will have 15 minutes of live questions and answers with Dawne. Throughout the webinar, feel free to use the Q&A box on the left-hand side of your screen in order to submit your ddPCR questions directly to us. We will get to as many of your questions as we have time for. However, if we cannot get to your question today, we will personally follow up with you over the next few weeks. Also, if you have any technical difficulties during the webinar, please use the Q&A box to report them. Now, a little bit about our speaker, Dr. Dawne Shelton. Dr. Shelton received her Bachelor of Science degree in Biochemistry and Molecular Biology from the University of California, Santa Cruz. After 5 years working in the biotech industry at Geron Corporation, she pursued a PhD in oncological sciences from the University of Utah Huntsman Cancer Institute. Here she investigated different aspects of the Wnt pathway in colon cancer. Dr. Shelton then went on to study the origins of the different subtypes of breast cancer by creating mouse models within the context of each cellular lineage. She moved to the University of Iowa and examined uterine cancer in a clinical cancer research lab. She is now a senior scientist at the Digital Biology Center at Bio-Rad, focusing on developing applications and collaborations for ddPCR in the field of cancer. The title of her talk this morning is Droplet Digital PCR Applications for Molecular Pathology and Liquid Biopsy. Without further ado, let's get started. Dawne?
Thanks, Mohini. Hello, my name is Dawne Shelton, and I'm a Senior Applications Scientist at the Digital Biology Center in Pleasanton, California. I was hired to help translate the new Droplet Digital PCR technology into applications in the cancer field. I'm here today to talk to you about the molecular pathology and the liquid biopsy with ddPCR. Droplet Digital PCR is the third generation in the evolution of PCR, moving from qualitative to absolute quantitative measurement. In real-time PCR, the effects of inhibition either by primer efficiencies, quality of sample, or chemical inhibitors are always a concern. Any cause for delays in CQ values alters fundamentally the accurate measurement of the template, leading to greater errors. With Droplet Digital PCR, each droplet is thermocycled to endpoint individually and read as either a positive or a negative for the target of interest, hence the digital aspect of ddPCR. ddPCR enables highly precise and sensitive nucleic acid quantification in a convenient, high-throughput manner. We take a typical 20 microliter reaction and partition it into about 20,000 droplets. Run the plate in a standard 96-well thermocycler, then read each droplet in a flow cytometer-like fashion. Droplets containing the target become fluorescent and are counted positive by the droplet reader. Droplets without target are negative. For one reaction effectively goes from analog to thousands of discrete yes-no answers. The key technical advantages of ddPCR are summarized here. First, as each droplet is a yes-no answer for your target, ddPCR simply counts the number of input molecules. Your output, in copies per microliter, tells you how much amplifiable DNA you screened. There is no standard curve, and because we read at endpoint, there is minimal worry about PCR efficiency. This is especially important on degraded clinical samples. Second, a digital readout of 20,000 droplets gives high precision and reproducibility, allowing us the ability to discriminate small differences in nucleic acid content. This high precision leads to high sensitivity, which enables the detection of rare events like cancer mutations or virus in plasma. In effect, through sample partitioning, droplets serve to titrate out the high copy backgrounds. effectively increasing the signal-to-noise ratio of a rare target. In this example, the bulk sample has a mutant species present at 40 mutant copies in 40,000 wild-type molecules. When the sample is partitioned into 20,000 droplets, 40 droplets will contain the single mutant target, now at a ratio of 1 in 3. This year, we have brought new products to market to meet the needs of our growing customer base, the cancer researchers. We've recently released the AutoDG, a machine that automates droplet generation up to 96 samples at a time. There is minimal hands-on time, a high throughput format, and consistent expert quality droplets from any user. We've also been expanding our catalog of wet lab validated prime PCR mutation detection and copy number variation assays optimized for the ddPCR platform. We now have assays that target the top mutations or genes present in Cosmic, with thousands of expertly designed in silico assays for the less prevalent targets available today. This is an example of our validation data for one of our most popular mutation detection assays for BRAF V600E. For our mutation detection assays, we use a single primer pair to amplify the mutation site, then 2 competitive probes to detect mutant or wild-type copies. Mutant is in the FAM channel, while wild-type is in hex. Shown here is an example of the wild-type only control well in the upper left-hand corner and a mutant-positive control well at 0.1% in the upper right. The 2D plots shown here display each droplet in the reaction. The no-template droplets are in gray, the wild-type single positives are in green, and the mutant-positive droplets are in blue, with the mutant and wild-type double positives shown in brown. The limit of detection plot below shows a 0.1% mutant detection in a single well. That's 1 in 1,000, with clear separation from the wild-type only negative control as indicated by the dashed line. This is the same assay with a 10-fold lower LOD in 4 merged wells. Each of our validated assays are tested to 0.1% in a single well, but all can achieve much greater sensitivity by the addition of more wells, more target DNA, and the merging of data. In addition, one of the main strengths of ddPCR is the precision counting of input molecules, which means 3 users with 3 instruments and 2 different locations produced near identical results from 1% to 0.01% as shown here, both as a percentage mutant as well as an absolute concentration on the right side. So just to get an idea of what kind of samples our customers are using, we'd like you to fill out the questionnaire here. What sample do you most commonly use? Please select an answer and push the Submit button. So it looks like most of the people watching today are using cell-free DNA, some formalin-fixed paraffin-embedded tissue, and some version of other. Today's webinar will focus a lot on cell-free DNA. So the liquid biopsy is a holy grail in diagnostics since it is largely non-invasive and believed to be more representative of the overall body, rather than a biopsy of a tumor in a single location. In addition, multiple biopsies over time are considered impractical, expensive, and can be extremely dangerous for the patient. However, there is very little DNA in plasma, about 5 to 50 nanograms per mil in advanced cancers. And the quality of the DNA is very poor, mostly apoptotic fragments. Most of the DNA is also wild-type in nature, arising from non-tumorigenic cells. These limitations have prevented the use of plasma DNA as a diagnostic test up until very recently. Shown here is data from the lab of Luis Diaz at Johns Hopkins University. In this figure, you can see the percentage of patient samples in the y-axis that have detectable cell-free DNA in the plasma from different tumor types in advanced disease, shown on the x-axis, indicating that greater than 80% of patients with metastatic disease have detectable levels of cell-free DNA. The figure on the right indicates the number of copies detectable per 5 mL of plasma for each of the tumor types. They further go on to show that the levels of mutant cell-free DNA vary by both stage of disease and location and can provide therapeutic, predictive, and prognostic information, making it promising for monitoring residual disease. Today, the use of tumor genotype to guide patient treatment is routine in a variety of cancers, including melanoma, colorectal, and breast cancer. Roughly 50% of the melanomas have activating mutations in BRAF, and V600E in particular accounts for an estimated 80 to 90% of that half. Personalized cancer treatment of the future in melanoma requires detection of activating BRAF mutations. So we formed a collaboration with Dr. David Polsky at the New York University Langone Cancer Center to evaluate the potential of digital PCR to measure cell-free DNA from the plasma of patients undergoing targeted therapy or immune checkpoint blockade in melanoma. They evaluated 115 samples from 25 patients over the treatment course. Initially, several platforms were tested, and ddPCR performed exceptionally at detecting a single mutant copy across the range of DNA input amounts when a sufficient number of replicates were run to overcome stochastic distribution effects. Shown here are 3 DNA dilution series at 300 nanograms, 30 nanograms, and 3 nanograms DNA with 10-fold dilutions ranging from 10% to 0.01%. ddPCR was able to resolve a single copy at 0.01% in this experiment. This is an example of one patient from the study whose mutant BRAF levels tracked with the progression of disease. Shown here is their response to BRAF and MEK inhibitor treatment, stable disease for a short time, and then rising mutant DNA levels with the disease progression. For this patient, rising cell-free DNA levels were detected 43 days prior to standard clinical detection, with a study average of 84 days prior to scans. In 92% of plasma samples, changes in DNA levels trended with disease status as evidenced by radiographic scans. Though much work still needs to be done, this studies and other like it show promise for monitoring residual disease and recurrence using ddPCR. EGFR signaling is a significant driver of metastatic colorectal cancer, and blocking EGFR signaling is one of the major treatment options. However, downstream activating mutations in KRAS block the benefits of anti-EGF therapy in colorectal cancer in about 40% of colorectal cancer cases, and are therefore counterindicated for anti-EGF therapy. There are 7 mutations present at greater than 1% population frequency, and an additional 15 more present at less than 1% frequency in cosmic. To address the need for screening a greater number of mutations with a very limited biological sample, we developed the KRAS screening multiplex. This assay is designed to screen the 7 most common KRAS activating mutations in colorectal cancer samples. It has 1 set of primers, 1 wild-type probe in hex, and 7 mutant probes, each in FAM, covering the most common G12 and G13 mutations. The 2D plot above shows an example of a positive control for the screening assay using the 7 mutation mixed positive control sample. This concentration plot represents the validation data for the KRAS multiplex assay. KRAS mutant DNA, indicated in blue, was run in a 3-fold dilution series in a constant background of about 40 nanograms human wild-type genomic DNA, shown in green. This established a sensitive single-well detection of KRAS mutations down to 0.2%. The plot on the right shows how sensitivity is a function of the percent mutant and the number of amplifiable copies screened. This is an ideal world example of reference to be used as a reference point since it assumes perfect DNA sample, a perfect assay, and no false positives. So for future development, we are interested in knowing what screening multiplexes do you wish to screen for. Please choose all that apply. I'll wait a few minutes, and then we will gather your results. Oh dear. Most people seem to want other. Okay. So KRAS G12 and G13 mutations, about 25% of our respondents. PIK3CA, we will talk about that today, as well as BRAF V600. Thanks for your participation. So these are the 2D plots for the KRAS screening multiplex assay on cell line DNA, FFPE, and cell-free DNA. As you can see, there is a fundamental difference in the plots representative of the quality of the DNA sample being inputted. Cell line DNA gives clear separation of the 4 clusters as shown on the left. while highly damaged FFP tends to have intermediate fluorescence droplets, or RAIN, due to the presence of non-amplifiable DNA and PCR inhibitors, while apoptotic cell-free DNA tends to have an intermediate phenotype, as shown on the right. These are the concentration plots and fractional abundance graphs for the percentage mutant of wild-type KRAS in green, mutant KRAS in blue, and the percentage mutant in orange from 6 MD Anderson Cancer Center cell-free DNA plasma samples, demonstrating the range of mutant detection in real samples. We take a fairly conservative approach towards calling a sample positive based on non-overlapping 95% confidence interval error bars between the appropriate wild-type control and the samples. For instance, sample 1 falls below the dashed line, while sample 2 falls above. We call sample 1 a negative for the KRAS G12A, C, D, R, S, V, and G13D, and sample 2 and above positive for at least one of those mutations. This is data from Helen Huang's very first experiment showing excellent concordance between the CLIA-tested tissue samples and ddPCR from plasma, both in duplex and multiplex assay formats. The apparent discrepant sample, number 1, was actually a mutation not present in the screening assay, Q61H. Therefore, it showed up correctly as a wild type for the 7 KRAS mutations screened. For in-house testing of the KRAS screening multiplex, we purchased plasma samples from Conversant Bio, representing 12 patients and 52 draws, with 5 patients testing KRAS positive by CLIA tissue testing at diagnosis. We also purchased normal samples from ProMed DX, representing 12 pregnant patient samples. We used the QIAGEN circulating nucleic acid kit to process these samples. This is the first graph. Cell-free DNA was quantified by using duplexed copy number assays on the 12 normal and 12 cancer patient samples, a total of 64 draws. Using ddPCR, it is clear that the amount of amplifiable cell-free DNA is highly variable, both across groups and across draws for a given patient. The median value of cell-free DNA for the normal samples was 3.8 nanograms, while the KRAS mutant samples had a median value of about 30 and the KRAS wild types a median value of about 20. These numbers are concordant with other studies published. This patient had 15 draws over a 7-month period. As you can see, the amount of DNA per draw was highly variable. Unfortunately, we don't have any data to correlate these time points based on treatment or response. We then used the KRAS multiplex assay to detect KRAS mutations in normal and patient cell-free DNAs. The plasma samples were taken years post-diagnosis, and as such, we were not surprised to only find 1 KRAS positive patient sample. This was present at 1%. None of the normal or wild-type KRAS patients revealed a KRAS mutation using this strategy. Further analysis using individual duplex KRAS mutation assays identified the M3K patient sample as KRAS G13D. Wild-type controls were used with each assay to identify the correct false positive rate for each assay, and positive calls were made by non-overlapping 95% confidence interval error bars. Since we did not have enough sample left to screen all the individual mutations, and we had already determined the absolute concentration of the sample, we used the So Advanced Preamp Supermix from Bio-Rad to generate enough sample to identify the individual mutation type. There was some amplification bias, which altered 1% mutant to about 0.64%, as would be expected with preamp. But since we had an absolute measurement from the initial screen, a qualitative answer was acceptable. Another key advantage of ddPCR is the ability to identify PCR inhibition by the 1D or 2D plots. Shown here is a 1D plot with one sample loaded at 6 microliters and 2 microliters, where you can see the obvious suppression of the fluorescence amplitudes as indicated on the y-axis, representing PCR efficiency as compared to the controls, wild type. As long as there is clear separation between the droplet clusters, accurate quantification can be made. When separation is poor, the fractional abundance will generally still remain accurate. As you can see, our samples loaded at 6 microliters and at 2 microliters showed by back calculating about 150 to 160 copies per microliter of the stock. To further evaluate the performance of the KRAS screening kit, we also purchased colorectal cancer FFPE samples from Advanced Tissue Services. We used the QIAGEN DNA Easy kit to process the 16 KRAS mutant and 4 grossly normal samples. We first extracted the DNA from FFPE using standard protocols. We then used duplex copy number assays to estimate the fraction of sample that could be PCR amplified. 13 out of 20 colorectal cancer samples had less than 5% of amplifiable material. We found that the amount of amplifiable material is not linearly related to the amount of DNA detected by nanodrop. For example, several samples with greater than 100 nanograms material by nanodrop had less than 10 nanograms amplifiable material, which has significant implications for the interpretation of data and sensitivities of genomic tests. We have found that the yield of amplifiable DNA from FFPE samples is highly variable and not well assessed by spectrophotometric analysis. Shown here are measurements made of the Horizon FFPE reference standards for each of the 7 KRAS mutations measured by the specific Duplex assay or the KRAS Multiplex. Here we demonstrate quantitative measurement of FFPE using both the Duplex and Multiplex assays. As you can see, the concentrations are near identical with overlapping 95% confidence intervals. We use the KRAS multiplex screening assay to detect the 7 most common KRAS mutations. 6 of those samples had a mutational fractional abundance of less than 3%. We also use the 7 individual KRAS mutation assays to identify the dominant or sole KRAS mutation picked up by the initial screen. Example M2K shows 2% mutant, and the single assay identified that mutant as G12D. Our data was consistent with COSMIC. The majority of KRAS mutations, greater than 80%, in these colorectal cancer samples contained either G12D, G12V, G13D, or G12S. Further, similar to the Horizon references, measurements of FFPE samples by duplex and multiplex assays correlated strongly with an R-squared of greater than 0.99. As mentioned previously, you can use the 1D and 2D plots information to visualize and troubleshoot your experiment for PCR inhibition. On the left, the sample was loaded with 5 microliters of input, and on the right, 2 microliters. 20% of this material was amplifiable, but the vast amount, 80% or greater, was not, as well as there being inhibitors present. This impacts the positive amplitudes in both the FAM and HEX directions. Loading less sample or diluting your sample out allows for better amplification and accurate quantification. So another thing that we're finding to be very important as this process moves forward is the sample extraction method that you use is critical. So what we wanted to know here is what sample extraction methods do you use? Please choose all that apply. Okay, it looks like most people are using the QIAGEN kits. I'm going to, for the DNA Easy blood and tissue, blood and cell culture, and the QIAMP circulating nucleic acids kits. Excellent. Another field of study quickly being adopted with the advent of whole genome data is the study of copy number alterations or variations. Roughly 12% of the genome has been found to be copy number variable in the normal population. These variations may be caused by duplications, deletions, inversions, translocations, or non-disjunction events. In addition to small regional changes, entire chromosomes may be deleted or duplicated in disease states. Germline variations might lead to susceptibility in disease or adverse reactions to certain drugs. In cases post-onset of cancer, the cancer process itself acts as an accelerated genetic selection process. This somatic expansion and selection process results in a survival of the fittest cancer cell race, leading to many somatic copy number alterations and genomic imbalances. The implications of these variations are not currently well understood. However, one well-documented example would be the amplification of HER2 in breast cancer. Studying copy number variation in cancer is not a trivial exercise. For ddPCR, we have found it requires short, matched amplicons for your target and reference. When using damaged DNA types like FFPE or cell-free DNA, the smaller the assay footprint, the more fragmented DNA you can detect. Shown here are two assays against MYC, one for cell line DNA and the other designed for fragmented samples. When the reference gene and the target are not matched, you can get significant alterations in your CNV number. In this case, a copy number 1 versus a copy number 2 difference. Tumor heterogeneity also causes problems for copy number variation studies. Identifying the most appropriate reference for your tumor type becomes the most critical issue. We recommend using multiple references and multiplexing. Normal population references, especially TERT, are not appropriate for many cancers, as they are frequently deleted or amplified in certain cancer types. One thing that you can do is check on the COSMIC site and for your references of interest and your cancer subtype of interest, and to identify targets and regions in the chromosomes that are least likely to be deleted or amplified. Due to the heterogeneous nature of tumors, a single reference is often inadequate for calling copy number. Shown here are 4 cancer cell lines tested for HER2 with 2 different references. In the first 2 cell lines, the HER2 copy number is identical for those cell lines. However, SKN-MC and HS578T, for those 2 samples, the choice of reference alters the copy number call. To address this, we have wet lab validated about 60 periocentromeric reference assays corresponding to each chromosome. Whatever your cancer subtype of interest is, you can find a validated reference assay for it. For a blind approach, we asked our bioinformatics group to figure out a method to determine the average genome equivalence of a sample, or a baseline reference number. They determined that with 8 references, at least 4 will be informative 99% of the time. By tossing out the outliers, shown in red circles, and keeping the middle 50% inner quartile range, you could establish a good reference number for your target of interest. Using this method, we tested 6 colorectal cancer cell lines and 4 targets, MYC, EGFR, SMAD4, and TP53. We then compared our results with COSMIC data. ddPCR and the SNP array data in COSMIC gave high correlation with R-squared values of greater than 0.98. The exceptions were actually copy number states higher than 10, where the dynamic range of the SNP arrays showed suppression. For example, shown on the right are the MYC copy numbers, where COSMIC data suggests a copy number of 14, and ddPCR measures about 10-fold higher. We saw this in multiple different instances. In this recent publication, Jeffrey Oxnard and colleagues at the Dana-Farber Cancer Institute used ddPCR to monitor patient treatment in lung cancer. These researchers wanted to use cell-free DNA from patient plasma to track and detect EGFR sensitizing and drug-resistance lung cancer mutations, such as T790M, over the course of treatment. To evaluate this possibility, they performed rare mutation detection of multiple EGFR, KRAS, and BRAF mutations on cell-free DNA from lung and skin cancer patients. Here I'll highlight the work on EGFR in lung cancer. First, they used the conventional assay validation approaches to establish the sensitive detection of EGFR L858R and T790M mutations down to less than 1 in 10,000 copies using digital PCR. They then applied these assays to plasma from lung cancer patients undergoing therapies. One example of this profiling is shown. You are able to see serial non-invasive mutation detection during treatment, including the rise of mutational loads as the brain metastases evolve. In another compelling finding, the team was able to show using digital PCR in 6 out of 9 patients the reemergence of EGFR mutations detectable up to 4 months ahead of objective disease progression, or PD, by radiographic imaging. This sensitivity has the potential for massive patient benefits, both in terms of comfort, safety, and the ability to quickly iterate on the treatment options. This is a paper published last February. What is remarkable about this, aside from detecting mutations in plasma, is that it is the first study to examine early-stage disease using digital PCR successfully. All previous studies have focused on late or metastatic disease because there is so much more circulating DNA available. Our collaborator at Johns Hopkins was interested in studying plasma markers in early-stage breast cancer patients, focusing on the PIK3CA mutations. Typically, patients with early-stage disease have surgery to remove the tumor, then chemotherapy. However, 60 to 70% of patients that are treated with chemotherapy when surgery alone would have cured them. This investigator would like to identify those patients. So 29 patients were enrolled in a prospective study. The clinicians took both preoperative blood samples and postoperative blood samples, in addition to FFPE samples of the excised tumors. The tumors were sequenced by Sanger sequencing, and 10 patients were found to have PIK3CA mutations. However, when digital PCR was applied to those exact same samples, an additional 5 patients were found to have PIK3CA mutations. This was due to the greater sensitivity of ddPCR. When digital PCR was applied to the plasma samples, 14 samples were again found to be mutant, closely paralleling the FFPE data. Several weeks after surgery, 5 patients continued to show mutant DNA in their plasma, while 12 did not. This meant that in this small prospective study, that ddPCR had 93.3% sensitivity and 100% specificity. Since our first publication of digital PCR in 2011, our customers and collaborators have found hundreds of ways to use ddPCR in their research. This speaks mainly to the ease of use and affordability of the system, as well as to the technical advantage of ddPCR. Our applications group, as well as our customers, are putting ddPCR to use in multiple areas of cancer research. This includes rare mutation detection and somatic copy number alterations that we discussed today. It also involves quantifying rare cells like circulating tumor cells, tumor-infiltrating leukocytes, and other applications already published are in using testing for microRNAs, methylation quantification, as well as enzyme activity, for instance, the telomerase telomere repeat amplification, or TRAP assay. For more information, I encourage you to watch our previous webinar on ddPCR cancer applications, as well as our webinars on CNV, gene expression, multiplexing, and tips and tricks. These are a few of the cancer publications you can check out from previous years for your reference. Thank you for your attention, and I'd like to open the talk for questions.
Dawne, thank you very much for that great talk. To the audience, if you would like a copy of today's PowerPoint presentation, publications list, the KRAS applications note, or a copy of the QX200 ddPCR brochure, they are available at the link to the left of your screen for download. The webinar is also available on demand within 24 hours. If you would like any additional information, please feel free to reach out to your local Bio-Rad Droplet Digital PCR specialist as well. Now for questions for Dawne. You mentioned copy number and mutation detection assays for digital PCR. Do you have any gene expression assays available for digital PCR?
Yes. Starting in January, we'll be selling gene expression assays on the Bio-Rad Prime PCR website.
And a follow-up to that, are these just probe assays, or are they primer assays available as well?
Primer, evergreen, as well as probe assays.
Thank you. During your talk, you mentioned sensitivity can be increased by combining wells. Is there a limit in your experience on how many wells you can combine, or are there any other approaches available as well?
For right now, the number of wells in theory that can combine is the number of wells that's on one 96-well plate. Now, for practical purposes, I'm not sure there are a lot of applications where you'd want to do 96 wells, but it's certainly possible. In the future, we'll have the ability to merge wells from across places.
Okay, so going back to the KRAS multiplex assay, is there a way we can validate for individual assays within the multiplex?
So we offer each of the assays that are in the multiplex are sold as individual duplex assays on the Bio-Rad site. Those assays are also pre-validated wet lab bench. You can download all the validation data straight from the website.
During your talk, you mentioned some sample extraction methods. In your experience, are there particular methods that give like more yield than others? Any other guidance surrounding sample extraction that we need to take into consideration that will have an impact on the digital PCR assays results?
Good question. So sample prep is turned out to be a bit of a concern, I think, for everybody, actually. For the care that needs to be taken when using different sample prep methods is actually the more important factor. So things like organics, phenol chloroform, and excess amounts of ethanol, for instance, left over from any of the sample prep methods. If those kind of organics get into your digital PCR, you can have disruption of your droplets. Obviously, this oil and water emulsion system doesn't like the addition of organics like phenol and chloroform. As long as they're cleaned out properly, then it's fine. The other thing to keep in mind is Droplet Digital PCR droplets are made in a cartridge that has microfluidics. Microfluidics don't like it when small fragments of schmutz, of small pieces of plastic, or beads, if you use beads in your cleanup methodology. Those small pieces of solid material will then plug your microfluidics channel. So that's something to keep in mind. All of the methods so far work fairly well as long as care is taken in the actual process.
Thank you, Dawne. So continuing along the theme of sample prep, we have a question regarding low yield of cell-free DNA. And in your experience, what kind of DNA extraction kit have you found to be good for digital PCR?
So there's been several papers out actually addressing these kinds of questions. I'd have to look up the references again, but Jim Huggett and the group at LGC has done a few papers like this, as well as a few other people. The QIAGEN DNA Easy for virus works quite well. The circulating nucleic acids kit from QIAGEN also works well. We've had excellent use with the Maxwell. We've heard excellent use with the Maxwell Promega kit. The biggest thing for when you're doing cell-free DNA is to optimize your protocol for smaller fragment DNA, because most of it is apoptotic fragments.
Speaking of fragments, is there a way to use the amplicons generated by Droplet Digital PCR for Sanger sequencing?
Yes, actually people have done that. We don't offer right now a way of sorting droplets. It's something we've talked about with various customers, and some customers are doing it on their own, some coming up with their own methods or using spectrophotometric methods. But we offer several droplet making protocols where you can pull out the droplets, the templates from the droplets, and sequence those.
Well, thank you very much, Dawne. Seems like we have no more questions. Thank you to the audience for attending Bio-Rad's Droplet Digital PCR webinar today. We have more webinars in the coming months, so please continue to participate. As a reminder, today's webinar will also be available on demand within the next 24 hours. For those of you who had questions that come up after this webinar, we will follow up with you personally in the next few weeks. For more information on Droplet Digital PCR and educational resources, visit www.biorad.com/webinar. This URL is displayed to the left of the slide in the resources box. Thank you, and have a great rest of the day.